All right, so got to know about the structure of DNA and RNA, so that's what we're going to do here. So you've all heard of A, C, T, and G being the main nucleotides. We got to get into what the structure is like. So when you look at a DNA strand, you see the sugars connected to the phosphates along the outside, and then these nitrogen bases are on the inside facing each other. We'll get to that in a little bit, how that works. But this A over here would join with a T on the other side. The C would join with a G. Then you'd have the other base pairs. So let's go ahead and label this nucleotide here. This is the phosphate group. Over here is the nitrogenous base. And down here is the sugar. And for DNA, this would be a deoxyribose. Deoxyribose. And you can tell it's deoxyribose if you have an OH and then an H. And we'll get to that in a second. OH here and then a regular H. So you need to be able to draw that, be able to identify those different structures on the IB exam. So getting into more of the differences between DNA and RNA. It's very similar, but also they got some important differences. In our DNA, we're looking at an OH here, and then just a single hydrogen atom there. In RNA, we have both of these being OH groups. So, yeah, you just got to memorize that. I'll think of some kind of mnemonic device. Maybe some of you can send me ideas for that. The number of strands is different. So, DNA is typically double-stranded, well, is, and then RNA single-stranded, typically. So we have two anti-parallel. That's like a two-way street. Been on two-way streets before. There's also one-way streets. And that would be more like RNA. But anti-parallel two strands, DNA or RNA that is single-stranded. Usually linear, but it can be folded up. And our bases, look, they have A, G, C in common, but DNA has thymine and RNA has uracil. So the T and the U switch. All right, that's the main thing. So T and the U, T versus U, and this comes to strands, 2 versus 1. And the type of sugar, deoxyribose versus ribose. Okay, when it comes to those sugars. And here we get this, it kind of gives you a good graphic, shows the anti-parallel. Look carefully, see this sugar is shaped like a pentagon. We'll see this, we'll say this is in pointing up. If you look over here, you'll see it's flipped the other way. So you have the nitrogen base with the phosphate connected to the left. Over here, it's just reversed. It's flipped upside down. So the connected to the top left is the lower right. And we have the nitrogen bases in the middle. That's all these colored blocks. And then connecting them together are hydrogen bonds. Or you'll see it H bonds. Not H bombs, H bonds. So, like we saw in that first graphic, sugar phosphate backbone is on the outside. You'll see here, the outside of your molecule. And the bases are on the inside. We got our nitrogen bases all lined up in the middle. And the base pairing rules, complementary base pairing, G and C. There's always 3 bonds in between those. And then A to T, and there's always 2 bonds in between those. and they go in, like I mentioned, the opposite direction. We'll say this is up, this is arbitrary, this is down. It's called anti-parallel. And you'll see once it actually is in the body, well, I mean, this is a simplified diagram here, but it'll twist up, forming what we call a double helix. What has been termed a double helix. So they took, or not they, Rosalind Franklin, she was one of the most critical people who came up with what the structure was like. X-ray crystallography, so very complicated, very tedious technique. You're making crystals out of nucleic acids and then running them through an X-ray machine and hoping to get some kind of an image that you can then analyze. And this was a critical image that she obtained. And by getting the distances on this image and then these marks here, they were able to measure the size of DNA and how it turns, where the nucleotides, where the bases are positioned, where the backbone is positioned. They knew the size of the phosphate, the sugar, and the nitrogen base, and they were able to fit it all together and come up with this model right here. And then remember, that's based from their knowledge of the size of these molecules, sugar, phosphate, nitrogen base, plus the X-ray crystallography data. And this is actually, I think this is pretty cool. I just found this diagram, or this picture of one of the actual first models they actually made. So the first model, they used cardboard because they couldn't get all these. They had a shop make all these things, but when they did, after the cardboard, they made this. Okay, once they figured out they had it. Pretty cool. So what you need to do is draw these for yourself. So maybe we'll go to this next page. You'll need to draw. This is my my sugar phosphate. Let me draw another sugar down here. Still don't know why that keeps popping up like that. I figured that out. So those, I got 3 sugars. I'm going to connect. See this bottom lower left of that sugar, that bonds to that phosphate. And this corner right here of the sugar bonds to that phosphate backbone. I'm going to draw, label that a P right here. I'm just going to erase it, but let's leave it there. So I got my sugar phosphate backbone. And to make it easier, I'm going to go ahead over here and draw my sugars upside down. Just draw them upside down. If you have a piece of paper, which you should, for your notes, you can just flip the paper around. Now I'm doing an okay job drawing it upside down. There we go. And we draw the phosphates here. I'll just draw the P right side up for my convenience. There. So I got, see how this is going this direction, and this is going, it's upside down, it's just flip. And then I'll just simplify this. I'll say these are my nitrogen bases. Say this is A and this is T. And there'd be 2 bonds there. Better connect it right there. So there's a bond. Toot, toot. And this would be a C, or a G and a C. 3 bonds. 2 more nitrogen bases. So just, do what I'm doing on your paper, and you'll get it. I'm going to go ahead and make this a T over here. Remember, they can be on either side, but it always has to be an A. Can't have 2 T's, can't have 2 G's, can't have a G and a T. So G to T, no. A to C, no. only A to T and G to C, or reversed. So T and A, 2 bonds in between them. So that's basically what you have to do. Draw that.